So that's sort of what got me excited to jump into the lysosomal storage disorder space. And I've been, learning ever since, trying to get up the expertise in the space. But there's a lot of opportunity to make a big difference. Like this therapy on patients in Canada, 5 patients treated. The patients are finding that they're able to stop doing enzyme replacement therapy. Huge difference in their lives. They're able to go from having to build their lives around clinical visits to suddenly they're able to live a normal life like the rest of us and go on long extended vacations without having to, constantly every 2 weeks go back into the clinic.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where our mission is to unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin. I'm a life science professional in bioanalysis from Kansas City. And along with me from North Carolina is my friend and co-host, Dr. Chad Briscoe. And Chad, he's a bioanalytical scientific leader from North Carolina. Here at BioTalk Unzipped, we bring experts from different corners of the pharmaceutical, biotech, and medical device worlds to share their innovations, insights, career journeys, and personal stories to the show. Please subscribe and thank you for listening. Today on BioTalk Unzipped, we're very excited. We're joined by Dr. Chris Hopkins. He's an experienced geneticist, biochemist, and entrepreneur. He's guided the acquisition of Nudra Transgenics by Vivo Biosystems. He participated in raising over $5 million in funding from venture capital, achieved more than $7 million in non-dilutive funding from SBIRs, licensed CRISPR technology in 2014, shortly after it was invented, and is the inventor on over 20 patents. He created more than 40 service product lines and cultivated a sales and marketing team that has achieved more than $10 million in market revenue. We're very excited to have you on the show, Chris. How are you?
Oh, doing fine. It's a pleasure to be here.
Well, great. I appreciate you being here as well. And it's part of our tradition here at BioTalk to give a $100 donation to just kind of as a thank you to the charity of our speaker's choice. And you've chosen the Environmental Defense Fund. Can you tell us a little bit about that organization and why it's important to you?
Well, it's one of my longtime charity choices. I'm an outdoor enthusiast. I absolutely love getting out hiking, skiing, mountain biking, and the encroachment on the free spaces. I want to be able to try and help mitigate that and try and make sure that we can all enjoy the resources that we all it's part of our national heritage. And I think that Environmental Defense Fund does a really good job of trying to keep those wild spaces wild and keep the access for the public to public lands.
Excellent.
Yeah, certainly a space that I'm passionate about too. So I really appreciate that, Chris. Thank you.
Sure.
Chris, you spent over 25 years in the gene editing and rare disease space, but Glafabra seems more, maybe more deeply personal and focused. Was there a moment or a conversation or something that really convinced you to build a company around enzyme deficiency diseases like Fabry?
Well, it stems kind of far back from, I guess my postdoc work. I was working on a gene called STXBP1. Actually, at that time, we called it UNC18. It was shortly after I left my postdoc, it was discovered that was involved in epilepsies. And as I was then getting into creating my first company, the one that called Nudra Transgenics, we were making humanized animal models in C. elegans and zebrafish. And those were platforms we were creating to do drug discovery for clients. Some of the stuff we've done was build like that very same genetic defect into those animal models using the human coding sequence. And we were able to then do some drug screens. And so I had a significant passion for the STXBP1. We were, trying to do drug screens there and try and find therapeutics that would reverse the phenotypic defects that were in the animals that were humanized. So that gave me a really big passion for that sort of preclinical drug discovery space. About a year ago, I left my CSO role at Nudra Transgenics. I grew that to 15 people. We were able to exit that one by acquisition into a new company called In Vivo Biosystems. In Vivo Biosystems has grown along quite well, doing a lot of CRO work, using those animal models as platforms. So shout out to that amazing team over there. But I decided to leave and do a therapeutics development because that's basically ended up sort of being one of my passions. I really wanted to be able to see it through, see those discoveries all the way into the clinic. And it turns out that with this particular one, I was talking with some academics at specifically Jeff Medine about some of the work they were doing, and I just sort of uncovered a interesting asset that was essentially academically developed in Canada, but was really ripe for bringing to the U.S. For an IND filing, so that we could get into the clinic. So that's sort of what got me excited to jump into the lysosomal storage disorder space, and I've been learning ever since, trying to get up the expertise in the space. But there's a lot of opportunity to make a big difference. Like this therapy on patients in Canada, 5 patients treated. The patients are finding that they're able to stop doing enzyme replacement therapy. Huge difference in their lives. They're able to go from having to build their lives around clinical visits to suddenly they're able to live a normal life like the rest of us and go on long extended vacations without having to constantly every 2 weeks go back into the clinic. Big difference in their lives, and potentially improving them for a better improvement in the symptoms than the current standard of care.
And how does it differ from, say, Amicus with megalostat or the gens? Well, it was Genzyme, I guess it's Sanofi now, but Sanofi, they did a lot in the ERT space, and I think also I'm also curious. Amicus, I believe, is moving into the gene therapy space with this also. So where's your sort of niche versus those existing treatments, or the development on the gene therapy from Amicus? Kind of curious about that.
Yeah, well, and let's sort of split that into sort of 2 buckets.
Sure, yeah, that's a few questions there. Yeah,
current standard of care. How do we differentiate? And the new emerging therapies, the gene therapies coming online. How are we part of or differentiated from them? So let's first tackle the standard of care. It's been amazing. The ability to take a recombinant enzyme made in the lab and now infuse it into patients has been dramatic. They no longer can die of kidney failure and don't need a kidney transplant, so they can endure and not need a kidney transplant. The other thing that also happens in Fabry is that the next sort of tissue that gets sort of poisoned by the buildup of toxic metabolites is the heart. And so heart failure is also sort of avoided with enzyme replacement therapy. So both you're stopping the progression of kidney disease and you're stopping the progression of heart disease by taking enzyme replacement therapy. So dramatically improving patients' lives. Yeah. But there's the CNS components of these diseases, of lysosomal storage disorders, that are very difficult for a protein infused into your bloodstream to get across the blood-brain barrier and to help mitigate the CNS symptoms. So that's one thing that our potential therapy can do a better job at. And you're getting a more continuous supply. Essentially what we do, to describe what we do, is we're taking the patient's own tissue, their own blood cells, the hematopoietic stem cells. We're taking those back to the lab. We're genetically engineering them so they'll express a protein. So we have a lenti vector that encodes a transgene that will make the protein that is missing in the patient. And now we have a bunch of cells that are repaired for their ability to make the enzyme. And we put those cells back into the patient as the therapy. So it's a cell therapy. Those cells, engraft in your bone marrow and then make white blood cells. Those white blood cells go all over your body and go just about every compartment in your body and be secreting that enzyme, which gets taken up by neighboring cells by a process of what's called cross correction. And then that leads to restoring the enzyme activity in your lysosomes. And you can get the essentially reductions in the buildup of toxic metabolites, so you can stop the progression of the disease. If you do it early enough, you could even consider it to be curative.
How early do you think you'll be able to do this in the future? Because if their screening is detected in a newborn, right? Can you do it at a week old?
When it comes to think about this technology, the backbone is very similar to CAR T. So CAR T is you're essentially doing the same thing. You're harvesting white blood cells or harvesting your hematopoietic stem cells, modifying them, putting them back into the patient. I don't know what the earliest CAR T is, but I know it's done in infants and children, right? So there is no reason why this can't be put in before the symptoms ever occur and stop you from ever having symptoms.
Yeah. Well, that's certainly if my child or family member or anyone was afflicted with it, I'd certainly want that to be the goal. So I definitely support that goal. One thing that I've wondered for a long time, I've had just coincidentally through the years, a lot of interaction with Amicus with different projects, and I'm curious why Fabry versus Gaucher or Pompeii, and they kind of landed on Fabry too, but I never really asked anybody there why Fabry. Is it an easier target? Is it a bigger population?
Well, yeah, and you mentioned the Amicus, so I actually didn't finish off I talked about the standard of care, and that reminds me to get to the emerging therapies.
Yeah.
Well, the big emerging therapy, like what Amicus is on, but actually one of the leaders in that space right now is Sangamo. They just cleared their phase 1, phase 2 fast track. And had brilliant results, really good effect, and they may be able to get a remat and skip phase 3 and just go straight to market.
Oh, nice.
That's what I understand they're working on right now.
Great for the patients.
Really, really amazing, and it's a very strong effect, just as strong as ours. But here's the drawback on about use of AAV, which stands for adeno-associated virus, right? So you take this virus, you put in the gene of interest into it, and now you use the virus as kind of this injection apparatus that is now going to hopefully have tropism to your or be designed to go to a particular type of tissue and inject that with the DNA cargo and lead to expression of your transgene. And so that's what those AAV therapies can do, and they can do it really well, probably and potentially stronger than you can get with the standard protocols. Enzyme replacement therapy. And the big problem is those things that are not perduring. I had one patient come reach out to me saying, the therapy has stopped sort of working in me. I had to go back on enzyme replacement therapy. And they told me I cannot go back on and get another dose of AAV. You can't get another dose of AAV because you got antibodies and you got one and done. Our therapy can be repeated as many times as needed. There's no reason to imagine that if we fizzle out after a little while, you can't get repeated. We have those 5 patients done. 4 out of 5 patients still to this day are really good control of their enzyme level. I mean, their toxic metabolism, metabolite levels are well under control. 4 out of 5. But there's that one patient, after 4 years, they fizzled out and they had to go back on enzyme replacement therapy. He's reached me and asked me, hey, can I get back on can I do another dose of your therapy? And there's absolutely no reason why I can't. There's no capsid around. It shouldn't be an antibody reaction. It absolutely should be doable. If we can get these things moved away from the myeloablation protocols, we currently use a submyeloablative protocol that doesn't require. It's an outpatient procedure, so it's a very mild conditioning protocol. But potentially, there might be angles to move away totally from any conditioning, or have it just be trivial. If we can move to T cells, that's another thing we're going to be trying to move to is working with T cells as essentially the manufacturing facility that makes the recombinant, essentially produces the enzyme that's missing, right? So that's that's the only way to think of our cells is they're essentially mini factories, little mini pharmacies, cruising around in the cell, spitting out and producing what's needed. And so that we can if we can utilize that kind of a system, we can have something that can be one-time treatment that can last many years, but is repeatable and can be done as many times as per need. And I think that's where our big distinction over the emerging therapies arises.
And the goal in I think probably in all CAR Ts is to try and be able to do that in vivo treatment instead of doing the manufacturing ex vivo. Is that a realistic or even a pipe dream for you guys that you might be able to go that route?
It's exciting to think about in vivo,
Yeah.
but now you're
Yeah.
basically just AAV with a larger cargo.
Yeah. Yeah.
And now you're probably going to have all the antibody
Yep.
challenges that you
Yep.
have with the AV. So a one
Yeah.
and done
Mm.
Yeah.
methodology.
Yeah.
We could maybe do that, and then if that
Yeah.
fails, go back to the original, which is
I
edit
suppose.
the
Yeah.
cells
Yeah.
and do
Yeah.
that. I think
Okay.
actually the production one that's most attractive is trying to figure out how to get to allogeneic sourcing.
Mm-hmm.
So,
Mm-hmm.
I mean, this is a challenge for patients. And
Yep.
in fact, patients probably do like autologous. So what is autologous? Autologous
Yep.
means we're taking your own cells, editing them or manipulating them
Mm-hmm.
with genetic modifications so that we're essentially reprogramming them or restoring them
Mm-hmm.
to the normal state.
Mm-hmm.
And then we're putting those back into you versus, hey, I'm going to go to Joe Blow down the street and grab their tissue
Yeah.
and squirt
Yeah.
it into you. Do you want that?
Yeah.
But if that one right now is, you might say yes, but you're going to have graft versus host disease challenges.
Yeah.
So you
Yeah.
have a big challenge there to come over with allogenic.
Yep.
But I think allogenic is making a lot of progress in
Mm-hmm.
that, wiping out the HLA loci. That's what Poseida is essentially why they got purchased by Roche.
Yeah.
I think it's Roche or Novartis. Anyway, there is a big push to be allogeneic, and I think that one actually would scale if the main route to clinic is one where it's kind of a shelvable asset, where it's almost part of a formulary. Currently, right now, the way these are done are very different. It's kind of it's definitely not on the formulary listed on the formulary. It's more of a when it's this personalized medicine, it's more of sort of at the choice of each institution's doctors to
Yep.
essentially execute this type of therapy.
Yeah. So the allogeneic would be basically an off the shelf, and I think it's
Potentially
kind of the way to look at
bankable,
it.
right?
Yeah,
So anywhere you
bankable,
can
and
kind of get
yeah.
bankable and just
Yep.
store it up.
Yep.
But
So
autologous is still not bad because
Yeah.
especially if you can get down to say like if you don't have to work with hematopoietic stem cells because to get hematopoietic stems to get them to engraft, they'll
Mhm.
naturally engraft back in again on a conditioned host. But if you don't get them back into the patient very quickly, they start differentiating very quickly. And
Right.
it's dependent on the media and so on.
Okay.
There's some tricks there that might improve. So if you can get them to keep them in
Yeah.
the A to C state for a longer time, you
Mhm.
might be able to then still get them to a graft. But they don't
Okay.
really bank well. But now if you can work with, say, B cells or T cells, I think T cells is the really exciting one. Those things can endure a long time in the patient. And you can grow them in the lab. So you can grow up a batch, take a fraction of the batch, use it for therapy, and freeze the other batch. And then if it fizzles, go back to the
Mhm.
freezer and
Okay.
unthaw and grow it up
Okay.
a little bit more again.
Yeah.
So you can have an unlimited supply for each patient.
All right, Chad, I think it's time for our first section of new news.
New news.
In Reuters, this was published just the other day. They published an article that said the FDA requests resubmission for rare disease therapy. And so they had requested that Stealth Biotherapeutics. When I first read that, I had to do a double take because it sort of threw me. So there's so many stealth companies out there, and there's a company also called Stealth Bio, right? So they asked Stealth Bio to resubmit its application for Lentiviral and experimental therapy targeting Barth syndrome, a rare genetic disorder with no approved treatments so far. Following the prolonged review, the FDA issued a complete response letter leading Stealth to reduce its workforce by 30% to conserve resources for that. Hopeful they'll get to that resubmission point. Chris, given the FDA's kind of cautious stance on accelerated approvals for therapies targeting ultra rare conditions like Barth, how do you navigate the regulatory pathways for Glafabra's treatments, especially when dealing with limited patient populations and the urgency of unmet medical needs?
Oh boy, that is a loaded one, right? Do you cut and run and go to some other country where you can get it all done and then try and bring it back in and be almost a medical tourism kind of thing? That's a crazy idea, though maybe not too crazy.
Yeah.
Um, and with the FDA cuts right now, that there's a huge debate out there of are they going to be able to keep their timelines, right?
Right.
So we're I, the gold standard in the world is to work with the FDA, and I think that we should do that, right? When you see this cautiousness, I think it's justified. We need to proceed with caution, but I also do think that we need to balance both there is the, oh my gosh, there is some patient harm that we could do right now. Let's be cautious. But guess what? On the flip side, if you don't allow these things to get out there when people could have been cured or saved,
Mhm.
and you're blocking it, and time will tell, right? You can kind of go back and sort of say, okay, look, you probably could do this even now. Say, okay, this finally got out and got released, but if it got released quicker, there were X amount of people who would have lived, right? Not yeah, I mean, it's very sad and very dangerous when
Mhm.
people get hurt in a trial. Okay,
Right.
we want that to be as minimal as possible, but the flip side of that is is that by going super slow and super cautious, there are people getting hurt anyway.
Mhm. Yeah.
Mhm.
Yeah.
Mhm.
Yeah. It's a fine line to ride, and it's always difficult to know where you are in that continuum. Um, and we'll see what happens as the months and weeks press on in terms of how these changes that we've seen in the administration are affecting the response rates and review rates of the FDA.
And as I go and stare at a clinical trial, I am left now. I was very solidly, oh, let's go the US path. But I'm just like, wait a second. It might not be the best way to get my therapy on in the
Yeah.
patient's hands.
Right.
Maybe. I mean, the Australia setups are pretty amazing. You look at that whole
Oh yeah.
landscape of the discounts and
Yeah.
the IND hurdle is it's different.
Yeah.
And it's definitely not, so there are other paths to getting patients
Mhm.
treated.
Mhm. And Chris is kind of related, but a little bit earlier up the stream than the clinical. Has your approach or philosophy changed at all with the FDA's recent announcement about utilizing more non-animal methods in preclinical for the advanced biologics?
I've been a big proponent for the 3 Rs, right? So that's
Hmm.
the reduce.
Yeah. Yeah.
I think it's great.
Hmm.
Will organoids be able to capture everything?
Yeah.
Probably not.
Yeah.
There it's it you know Can we reduce the amount? Part of why there's so many animals being done is those rules and regulations by FDA. Is, well, this is the way it's always been done. We've always
Yep.
killed 10 mice, 10 rats, 10 pigs,
Yep.
whatever. If they can sort of say, okay, maybe we don't need to do that much, we can reduce the
Yeah.
amount of
If
animals
yeah.
being used. I think that's going to be coming on. Eliminating is a much bigger bar. I don't think we'll ever eliminate.
But at least I think it gives a path. My first impression was it gives a path to say, hey, if I can show I can get just as good of information from a different path, it'll be accepted to use this organoid or organ on a chip or some AI model that's yet to be developed instead of killing the animals.
Well, I'm going to
So
put a
yeah.
plug in for in vivo biosystems, my former company.
Uh-huh.
I mean, we were humanizing animal models, creating the human gene in the zebrafish, and creating humanized fish and
Okay.
humanized C. elegans, and being able
Humanized
to,
fish. Okay.
yeah,
Yeah.
you
Yeah.
imagine this STXBP1, a neurotransmitter
Hmm.
that's important for synaptic vesicle release.
Yeah.
If you're missing it in a human, you're fully missing it. You're
Hmm.
homozygous. You're not viable. If you're missing it in C. elegans, you're incredibly uncoordinated. You can put the C. elegans gene, engineer it back in, the regular gene
Hmm.
back into C. elegans, and it crawls around just fine. What would happen when you put the human coding sequence, the exact human coding sequence to make the protein that's used in humans? Would you imagine it to be able to crawl around, or would it be highly compromised? Turns
Yeah.
out it rescues it to a very high degree. Like in liquid, you can't even tell them apart when they're swimming in liquid.
Hmm.
So
Huh.
human proteins in animal models can create platforms for discovery of human-specific interactions. So high throughput screening can be done in those formats.
Wow.
Fantastic.
Yeah. That's interesting. That's remarkable. Chris.
So there's a lot of room for alternative methods besides cute little mice and bunnies.
Well, and I think it's motivating a lot of people to think harder on it. So it's one of the things I've just joined an APS team that was formed to look at these things specifically and to start sort of consolidating all the different information and ideas, and so that almost be a clearinghouse. So people, if you don't know where to go to get information or whatever, we can kind of help consolidate that within the APS organization and help share and redistribute thoughts and ideas from member companies and vendors and everybody else. So I wrote down in Vivo Biosystems because one of the things we're looking for is people who are providing these types of services, so we can help help raise the level for everybody. So it's exciting.
That is. It's great.
Yeah.
Chris, in an earlier conversation we had, you had mentioned, you talked about medical tourism earlier in our conversation today, but we talked about that Montana law.
Oh, really interesting, isn't it?
Yeah. So for folks who aren't familiar with this, the Montana state legislature has passed a law that's basically allowing people to pay for therapies that have gotten through phase 1, but you don't have to be in the clinical trial. You can go in if a doctor's willing or clinic's willing to have this therapy. You can go in and pay for it yourself and get the therapy while it's still in experimental phase.
Can I clarify that?
Yeah,
a little
please,
bit? So
please do.
That law said that entities can come in and set up a clinic to do just that.
Okay.
That's the pretty interesting. Like, oh wow, we can go create a clinic in Montana, and we do medical tourism all around the country, within country, to Montana.
Right.
That's the kind of concept. Now, is FDA going to let that fly? I don't know.
Right. Yeah. That's that whole kind of state versus federal, combat that we get at times, right? So, but will this, represent maybe a new model for clinical adoption in the U.S.? We talked a little bit about, will the FDA accept it? Are there any other ethical or regulatory concerns that it raises for you? Or are you more, optimistic that this could be a really good opportunity for patients to get help that they need, and maybe for new data to be generated?
I'll tell you this. Okay, so I mean, it is a weird kind of gray area, but there's another component to sort of throw in this. That the FDA is already doing this. Guess with what? Nutraceuticals. Is it grass? Is it generally recognized as safe? Okay, what are your claims? Can you doing this? Is that clinic going to be able to say, I'm going to cure your disease? No, you're going to have to have some sort of different language if you're going to conform to the nutraceuticals format. But it isn't that different for the FDA in that regard, because it's what is the FDA's main purpose? Keep the population safe.
Right.
Keep them safe from snake oil that hurts them.
Yep. I guess Glafabra would be real hard to claim as a nutraceutical.
Yeah.
Good point.
Well, here's all natural, coming from your own self.
Exactly. Yeah. What's more natural than that?
Well,
you can take that one back to the board.
Yeah, right.
This knucklehead from a podcast suggested.
So maybe like, brilliant, go for it.
Does it make us money?
Right.
Yeah, it's
Right.
the board. It's the board. Okay.
And do you think if it works, if this gets signed into law and clinics start to pop up in Montana, do you think this will be a model for other states to follow suit? And who do you think might be next?
I bet there'll be a lot. I wouldn't be surprised. My home state, which is a very big nutraceutical state, doesn't Utah. I'm in Utah.
Right.
I wouldn't be surprised if they don't find that very interesting. If it's something that can two things: bring revenue to the state, okay, and then help people, and make and have a path for people to get access to medicines that might or might not help them, right? It's basically sort of getting you access to the clinical trial perhaps a little easier. I don't know. It's an interesting mantra, but one of the things is the cost. I mean, so right now it's very expensive, at least in our space, to get therapies developed, and the patient workup is a huge cost center on that one, right? So how do we get reimbursed as quickly as possible? So if you could charge for the experimental therapy, that would at least allow us to not have to be raising so much money.
Right. Right. I think that is and you mentioned that before. I think it's a very compelling argument and a very interesting idea that would change the shape of how funding these clinical trials may change and morph into in the relative future. So we'll see. I'm more on the optimistic side. I like hearing about these new creative ways of getting things done to get to the same endpoint rather than just, well, this is how it's always been done. As long as people aren't getting hurt and there is safety and we can all agree on how to go about it, I think there's a lot of upside to it, in my opinion. Yeah.
It is definitely something I'm keeping my eye on. I don't know if I'm going to be a first mover on the curve, right?
Right.
The innovator's dilemma. I'm probably on the other side of the chasm, but I'm still very interested in what's going on the front side of the chasm.
Absolutely. Somebody's going to do it. There's always somebody who wants to be first.
Well, the longevity groups, they're going to be jumping in there heavy.
Yeah, makes sense.
Yeah. And you had mentioned before that we're in a challenging time, right? It's sort of capital's in sort of a nuclear winter right now, and the biotech investment environment's not where we'd certainly like it to be. How are you navigating the psychological and strategic pressure of building your program during a capital constrained era?
Yeah, I don't know. It's tough, incredibly tough, because it's basically on fumes. Even getting my friends and family to throw in a bone is challenging. I myself, have basically stopped all charity. Sorry, my environmental defense fund, but you ain't getting my money right now. It's so it's I don't know how we do it. It's just whether most people just sort of say survive. Hunker down and just figure out how you can just sort of run on fumes for hopefully. There's a lot of indications right now that maybe by Q4 things will go back into the bowl, and we'll start going again.
It does seem like
I will say that right now, quite a few VCs and so on are saying we're not cutting checks till Q4, and some of them are saying probably not till the beginning of next year. And so that's how long you got to run on fumes by. At least that's what it seems like. Some people will be, the pros that have already gotten money and are really successful, they're going to be getting their cash. But those of us who are more in that sort of like, in a women of prayer kind of thing, like we don't have the greatest track record, but we really want to do it. We're having a very tough time getting checks.
Yeah, because I think there's a lot of money out there, but it's just everyone's sort of holding on to it. Is my impression. So
I got there. Yeah. I'm a member of Bullpen, and one of the things one of the guys that came on just Wednesday's chat was God, he hit some astronomical number, like it's 100 trillion or something is just sitting there on the sidelines. Exactly where you gather that all up from, but basically there's a lot of pent up money hiding in the banks that's not going anywhere.
Yeah.
Dang.
Yeah.
Wow. Well, on that note, it is time for new news. So about a month ago, on May 1st, there was an article that came out in the Times out of the UK called Gene Therapy Trial Saves Boy from Death Sentence. And there's a 4-year-old boy with leukocyte adhesion deficiency type 1, a rare and fatal immune disorder, which for him has been effectively cured through a pioneering gene therapy trial at Great Ormond Street Hospital. And the treatment involved modifying the child's own stem cells to express the missing protein necessary for a functioning immune system. And this breakthrough offers hope for similarly affected children and avoids risk associated with donor transplants. I love hearing these stories, Chris, and success stories like this highlight the transformative potential of gene therapies. How do such breakthroughs like that influence your approach and Glafaba's approach to developing treatments for rare diseases? And what lessons do you think can be drawn for the broader biotech community?
Well, when you bring up that specific example, that is basically, again, it's like what we're doing. I mean, we're very identical to that in the sense that we're taking CAR T therapy footprint, everything that what CAR T has done, but instead of making chimeric antigen, we're putting in a different a gene that is the missing protein in the patient, and then we put those cells back in, and they provide that. Then they secrete that missing protein, and it makes has the therapeutic effect that you're seeing there. I mean, there's not a whole lot of difference other than the fact it's just we're making one protein, they're making another protein,
Mhm.
so we're very similar in that. So that's how that. I mean, those are we've got those success stories. We got our 5 patients in Canada success story. There's multiple ones for these genetically engineered cell therapies. So that's how are we we're in the cell and gene therapy space, but we're not really quite a gene therapy.
Huh.
We're a genetically modified cell therapy, right? So
Right.
we kind of are a gene therapy because we're doing very similar to what AAV is doing. We're providing the missing genetic material that makes a proper protein, but our actual drug is not a virus. We're not infecting people. We're putting in cells, growing cells.
Excellent. And kind of looking down the road, once we get past this dearth of financial activity in the biotech space, and you're getting into some clinical success and things are moving. What is your long-term vision for scaling up your treatment and building Glafabra to help more people?
Yeah, that it is sort of like what's your next steps, right? So from how do we want to make a difference in the lives of patients? Well, I think that there is, you're actually kind of stepping into the areas that we might need a CDA for. So I'm going to be
Gotcha.
very
And yeah,
loosey goosey on the
right,
details.
right,
Yeah.
yeah.
But basically, can we treat other enzyme deficiencies?
Uh-huh.
I believe there's a lot of ways to tweak the secretory systems of cells to do just that. And then so there might be some tricks to engineer the cells so they'll secrete better. I have some ideas there. So then there's and then getting to allogeneic where we can sort of put them on the shelf, and those allogenics have no immune response into the patient. If we can really, if the field at large can essentially settle on a particular cell type, that we can do iPSCs that way, right? And engineer them so they're the perfect vehicle for essentially introducing these things that are missing in the patient, right? So one way to think of it is that they're almost like they're autologously derived symbionts that you engineered to create therapeutic effects. You could even imagine that cells could be secreting small molecules that are therapeutic. So there's a lot of how can you engineer cellular. I mean, they're even thinking about that and doing that in biotics, probiotics, right? So literally engineering bacteria so that they secrete the right milieu to give you a very happy gut. So it's already, and those are seeing approvals. So that's already kind of happening. But now, can you essentially utilize that secretory capacity from your own? Essentially have a cassette where you drop it into your cells. So the technology did is to insert these genes efficiently to grow the cell populations to the right cell types, the right differentiation. Like I'm going to put a plug in Trail Biosystems because or Biosciences, Trail Biosciences. I just got through chatting with them. Amazing what they're doing in terms of creating a really uniform differentiation. They can make HSCs, they can make all the different white blood cell types. They're trying to figure out a whole bunch of other stuff. So there's a lot of stuff that's kind of going on in the space.
Oh, that's exciting.
It's exciting. So yeah, so you would love to be a platform company
Yeah.
versus a product company.
Don't say
I
that word
guess.
right
Yeah.
now. It's
All
not
right.
in vogue. We use the same core technology to apply to different indications.
All right.
All right.
All right. All right. Well, I'll look forward to
I'm not a
seeing how
platform.
you. I'll look forward to watching how you apply your core technology to alternative indications, Chris.
Absolutely.
Yeah.
Excellent. Well, you mentioned we like to get to know our guests a little bit as well on a personal level. And you mentioned loving the outdoors. I do myself, even though I'm in Kansas City. It's not as known for the mountains and the beaches here. But I also get involved in some mountain biking, which is actually pretty legit in this area. There's a lot of enthusiasts, and they've got some great trails. What are some of your favorite outdoor activities to do?
Well, mountain bike riding is a big one, right?
It's fun.
I live up in
Love
Park
it.
City, Utah. So
Nice.
basically, I've got this one called Flying Dog. It's my backyard, right? It's basically I hop on the bike, and in literally about 1 minute, I'm hitting that trail
Awesome.
and
Cool.
bust that one out. Get my stroller running and try and get my best time ever. So
Yep.
so there's a bit of that, and then backcountry skiing here is it's getting crowded. Too many people out now, but because the amazing equipment's I've been doing it now for like 35 years,
Wow.
hitting the backcountry skiing. And initially it was on 3-pin bindings with leather boots.
Yep.
And it's just amazing where that's
Wow.
gotten to now.
Oh yeah. Yeah. Yeah.
So once
That's
I've
cool.
never done backcountry, so you ski down, and you got to climb back up on your own.
Well, you've seen a Post-it sticky note, right?
Mm-hmm.
So now imagine, it's called mohair is the original thing because it was actually hair of an animal. But they've got little fibers. They're pointing in one direction. So imagine a sticky note with a bunch of fibers on the non-sticky side, and you put that sticky note to the bottom of your skis, and now you can hike up on those fibers, and it allows you to go on a pretty steep angle.
Wow.
And now once you get to the top, they're called skins. You whip off your skins, tuck them up, click into the heels, rip down some turns, get down to the bottom, pop off your skis, put on the skins, zip on back up, and repeat.
Yeah.
Nice.
One of the things a lot of people don't know also, Gregory, is that every ski resort is at least everyone I know of is actually leased land. They don't really own all their land. So if you don't use, you're really just paying to use their lift. So you could actually go out there, and if you want to put on skins and climb up the mountain, and you see that, right? And some places you see that more than others. But if you want to put on skins and climb up the mountain, you can go right onto their property and just go for it. And,
somewhat, right?
Yeah,
And that's the sort of state by state. The avalanches are so bad in Utah that getting through the gates, they're allowed to essentially not allow you to go through the gates. So getting some side country now is no longer the full legitimacy of sanctioned side country is pretty hard to get these days. Do I still go side country? I definitely look over my shoulder a little bit and go check it out. But you also should know what you're doing because I will put in a plug for avalanche awareness. The Utah Avalanche is amazing. Great courses for learning how to use your beacons and so on, and how do you read snow and learn how to do it safe. And it takes a bit. I've set off actually quite a few big slides testing a slope, and I probably shouldn't even been bothering testing it because I knew that it was going to go. So you just got to have that knowledge and restraint.
Yep.
Excellent.
That's awesome. Well, the Fabry population needs you, so stay safe.
Yeah, please.
Stay safe out there, Chris.
Be careful.
Well done.
Yeah.
Another one of our traditions, Chris, is to take a question from a prior guest, and they give us a question to ask the next person who they don't know who it's going to be. So I'm going to hit you with this question and see what you think. What is the impact of decreased international trade on your industry?
It's just going to drive the cost of goods sold up, right? So we're so international for just anything produced, right? That whether it's China or any other, any other X outside of the US, right? So that stuff's just going to get more expensive, and it's going to get just painted back onto the consumer in the US, right? So our drugs are getting more expensive, and they're going to have to just pay more. I mean, that's what tariffs do.
I had a crazy thought. If I can imagine a scenario where, an autologous and the manufacturing is in Canada, right? Like, so you're drawing the sample in the US and you send it to Canada, and on the way back, do they say, hey, we got to put a 10% tariff on your own cells you're bringing back? Because I mean,
wow.
And that is actually something that is literally a mathematic I'm trying to figure out, right?
Yeah.
Mhm.
Is it? Okay.
You can, yeah. Thing is like imagine going to China or going to India, where you might be able to get the entire procedure done for 1/10 the cost.
Yep.
And yet you could probably put in if you do it right, you can put in the controls and everything that it's just the same quality, right?
Mhm.
That's hard to beat. I mean, maybe you do tax that medical tourism, because that's, I don't know. It's just it's a weird landscape these days in that regard. How do you, what? Yeah, it's a sticky subject, but how do you sort of, maintain your independence as a country when the world is so damn global right now? Sorry, I used bad language, but we are so darn global these days that it's really hard to separate all the parts of everything apart without essentially wrecking the system.
Yeah.
Interesting.
It's a challenge. It's a challenge, and it's even it's a challenge. We try and stay non-political on here, but it's hard not to step up alongside that a little bit. So appreciate you.
Well, I will say that when you go back to this raising money, that is why everybody's sitting on their capital right now. Things are just too questionable to deploy. Right? I don't blame them. I don't blame. I would be, if I had a similar amount of cash, I'd be stashed and not deploying it.
Yeah.
Right. Wait and see.
Yep.
Yeah. Now it's interesting you mentioned the price difference with healthcare, because I went to a cell and gene therapy conference a couple of months ago, and there was an Indian scientist. They had developed a CAR T therapy successfully in India, and
that's pretty amazing.
For that therapy, it costs about $30,000 for the treatment.
And normally CAR T in the U.S. is about $500,000, right? So it's more than an order of magnitude lower cost, and they're still, they're pricing it at a price that probably gets them a margin. So wow.
Yeah.
Could we ever do that in the U.S.? No.
Nope.
Doubt it.
There
you go. I mean, medical tourism again, right? So it's there
you go.
So
awesome. Well, Chris, thanks very much for joining us today and sharing your story and sharing what the Glafabra is up to. You certainly have two cheerleaders here that are cheering you on and really hoping that this becomes a reality and it can move forward into further stages of clinical development. And we'll certainly be championing that as best we can from our end.
Gregory and Chad, I really appreciate you letting me be on today. I always love, as you can tell, I just love chatting about the science that we're doing and what we're trying to do to help patients.
Awesome.
And we love it too. We learned so much from these podcasts that we've been doing, and that's the best part of it, is the learning that we get out of it. So thanks for giving us that opportunity.
Hopefully it's accurate information, and some of it is, some of it might not be, but hopefully you found it informative, because I actually really enjoy trying to share knowledge. I think we need to share knowledge around the world, because that makes the world a less confusing space.
For sure.
Absolutely. Yeah. No, it's good. And I know our audience will enjoy this conversation as well, and keep them more informed of new treatments and technologies. So good addition. Thank you.
Yeah.